Last week, we talked a lot about different types of DNA technologies. We went over examples of how PCR can be used to do a lot of different things and how it works. We also reviewed how electrophoresis works, different types of DNA fingerprinting using restriction enzymes and using repetitive repeats, a little bit of information about the differences between sequencing and microarrays and what those can be used for. And I told you we would go over CRISPR and GWAS studies today. I've decided that we already have enough things to talk about this semester, and we're pretty tight on time, so we're not going to talk about GWAS today. If you're curious about how GWASs work, I'm happy to talk to you about it outside of class, but it's not going to show up on your exam. So our focus today is going to be on CRISPR and transgenic technology. CRISPR is a type of transgenic technology, and I think it's important to understand how we do this to understand how important this is. We're also going to, from there, go into some ideas about ethics of using things like CRISPR, and that's going to bring us into thinking about genetic testing. So that's going to be our path for today. We're going to start by thinking about how we use DNA technology to create transgenic organisms. Then we're going to talk about different ways that we can do that, both naturally and in a laboratory. Some of the pros and cons of these different types of techniques, and why CRISPR is actually a big change in how we approach this. We're then going to talk a little bit about how we use CRISPR currently and the types of things that we have already successfully used it to do. Connect that to whether or not we should, and other things that become an issue for that. So let's start by thinking about this use of DNA technology. The most common uses for DNA technology are creating transgenic organisms, identifying genes associated with phenotypes, and genetic testing. We spent a lot of time last class talking about identifying genes and different ways we might do that, as well as basic things like PCR. It's important to remember that both of these other techniques, creating transgenic organisms and genetic testing, rely on everything we talked about last week. They just use them in slightly different ways. To get started with this, I first want to talk about what a transgenic organism is and why we might want to make one. I also want to talk about a few misconceptions about what this means and how useful this term actually is. Transgenic organisms are organisms where a DNA sequence is introduced into the genome that was not normally there. Sometimes people will say that it must come from a different species. But that's not necessarily the case. You can make transgenic organisms that only contain genes that are found normally in that species. But the change that you've made isn't normally there. We often use these types of organisms to do things like remove the expression of a gene or add the expression. We also might want to check whether or not a specific mutation causes a phenotype. This is a way that we can validate things that we find about our own diseases. We can disrupt or delete genes or parts of genes, and we can also do a lot of other things that you might not have thought about before. Most of the time when we think about transgenic organisms, we're thinking about giving the organism the ability to do something it otherwise couldn't. But that is only one of these possible things. The two most common misconceptions are that DNA has to be from a different species. Most of the time, that's not true. Instead, it's a synthetic version of a gene the organism already has, with changes made to it via PCR. For example, if you want to make a change to a gene you have that has a mutation that has a phenotype, if you were to fix that mutation, you could fix the phenotype. You wouldn't be introducing a new gene, you would be repairing one that already exists. But because you're using a synthetic piece of DNA to do that, you would be a transgenic organism. The other misconception is that transgenics is always unnatural. New DNA cannot be introduced naturally, but that isn't true either. Horizontal gene transfer is actually very common. This is transfer where different species exchange helpful genes, and it can occur in multicellular and unicellular organisms. And there are multiple ways this happens naturally in the environment. For example, we'll talk about two key ones: transposons, which are pieces of DNA that can jump around within a genome, and viruses, which are a natural source of many transgenic materials. This is because many viruses can insert themselves directly into host genomes permanently, which can disrupt gene expression. And when they remove themselves from the DNA, they can take genes from the original host with them to the new host. So let's talk about a few examples. Most of the time when we think about the transfer of genes, we're thinking about vertical transfer. Horizontal gene transfer refers to the movement of genetic material between organisms in ways that do not involve them being related. Vertical gene transfer is from parent to offspring, but horizontal gene transfer is not. Only the gene that is transferred and nothing else. This frequently happens when organisms live in close proximity to each other. For example, fungal resistance in wheat is actually an example of horizontal gene transfer. In this case, the fungus that lives alongside the wheat contains a gene, FHB7, that confers resistance to blight. Because it lives in close proximity to the wheat, occasionally some of the DNA from the fungus is integrated into cells of the wheat. This provides a benefit to the wheat by making it resistant to blight. Individuals that have this rare transfer are more able to reproduce than those that are not. This is because having access to this gene from a fungus protects you from the presence of blight, which you can see on the end of the negative wheat over here. This is a naturally occurring change. Nobody tried to force this to happen, and it was observed within a wheat field that some wheat was affected by blight and some wasn't. When research was done to figure out why, they found that the only change was the insertion of a gene from a specifically different organism. And when they searched for that organism, they determined it was this fungus. So how is that possible? There are three ways that cells can pick up genes from other cells that are not directly related to them: transformation, transduction, and conjugation. In transformation or transfection, new DNA is acquired from the environment. In this case, a raw piece of DNA that is not inside a cell is taken into the cell and is able to be incorporated into the host genome. The DNA in the environment can come from lots of places. For example, it could come from other organisms that live close by. When cells nearby die, all of their insides are released into the environment. This is a natural process. So while the fungus living inside the wheat does eventually die, it has a natural lifespan, some of the DNA released in that process can be taken up by the wheat cells. If it happens to then be incorporated into the DNA, now the wheat will express that gene. Transduction is the use of viruses or viral vectors in order to move DNA from one organism to another. In this case, the gene that we're following has been picked up in some capacity by a virus. When that virus then infects a new cell, that gene is transferred to the new cell, and if it's incorporated into its DNA, becomes part of its vertical lineage. In bacterial cells, there's an extra way that we can send things, and that's through conjugation. Conjugation occurs through direct cell contact. In this case, extra chromosomes, which are temporary chromosomes, can be traded from one organism to another. In bacteria, this is generally a plasmid. Since plasmids and other extra chromosomes don't contain essential genes, Losing them and moving them around doesn't kill or harm the original parent. This is a really quick way for bacteria to spread things like antibiotic resistance. If one type of bacteria has a plasmid containing resistance, it can give that plasmid to other types of bacteria living in the same host and will give resistance to everyone, regardless of whether or not it's their parent. This is one of the biggest problems with using antibiotics ineffectively. The only organisms that survive are the ones that contain the plasmid, meaning anyone left over can easily get that plasmid through conjugation. So there are three major ways that cells naturally take up DNA from other organisms. They can pick up free-floating DNA through transformation and transfection. They can receive DNA from a previous organism through viruses, or they can trade these extra chromosomes with each other. So now we're going to talk a little more specifically about moving DNA pieces, like viruses and transposons, and why those can cause big changes and move genes between organisms. One of the key natural ways that transgenics occur when we find them in nature is through virus movement. This happens because viruses are frequently capable of creating proviral DNA. Proviruses are able to integrate directly into the host genome. This means that the provirus will now be passed vertically from parent to child. And it creates a legacy of infection. This is one of the reasons why certain types of viruses, like retroviruses like HIV, are so difficult to cure. Because the virus is able to insert itself directly into your DNA, the only way to remove the virus is to kill every cell that it is integrated into. If you miss any of them, they can spread to other cells and will also spread to any children those cells have. Non-retroviruses also insert themselves, but at a lower frequency. For example, several cancers are caused by the insertion of viruses into specific cells. One of them is pretty famous. Anybody have an idea what it might be? A virus that causes cancer.
HPV?
Yes, HPV. So because viruses can insert themselves into the genome, some of them are more likely to insert themselves in locations that cause cancer than others. Because of HPV's sequence, it's very likely when it does insert that it disrupts the way cells grow, causing specifically cervical cancer. This is a big problem because it also means that it cannot be cured. The only way to remove it is to cut out all of the infected cells. In the case of HPV, what generally happens is that the virus is able to enter and replicate inside cells. It then goes dormant. If at any time the dormant version of the virus is integrated directly into the genome, this leads to cervical cancer. In fact, this integration is one of the leading causes of this specific cancer and is part of how we detect it in biopsies from the cervix. You can look for PCR within the DNA of the individual, specifically for this insertion. The other thing that can happen is that because viruses can insert themselves into the genome, they can reactivate. This is what we see with viruses that are also not curable, like chickenpox and herpes. For example, shingles is reactivation of the chickenpox virus. Each virus has a specific type of cell that it's able to replicate in and insert itself into. This is why reactivated viruses tend to have very different patterns of behavior than the original virus. For example, chickenpox tends to cause mostly skin issues and some pain and some, inflammatory stuff, but it's largely not a big deal as long as you get it as a child. Shingles looks very different. That's because chickenpox virus is only able to integrate into neurons. Even though chickenpox primarily appears to affect the skin and results in all of these blisters, it actually only replicates inside neurons. And once that happens and it goes dormant, the skin lesions go away. When it reactivates, it tends to form a pattern where it follows the dendrites of the neurons it is inside of. That's why it tends to affect specific locations in the body where those dendrites are present. And it also tends to follow a spiky dendritic pattern. So we know that viruses are able to integrate into genomes. We also know that retroviruses are able to remove themselves from genomes and transfer to new hosts. If they happen to land in the genome next to a gene, they can take that gene with them. Now the new host will have both the virus and the gene from the previous host. And that can be one way that these genes can get from one organism to another. Legacy viruses often result in things called transposons. Transposons are sequences of DNA that can jump to different locations within the genome. These jumps are capable of disrupting genes the sequence lands into or carrying local genes with them. Unlike a virus, they tend to be unable to transfer themselves because they don't have capsules or other ways to replicate. However, when the cells become close to one another, transposons can jump from one cell to another through pili and other bridges. For example, transposons that are located within the genome can go through a transcription process that allows them to jump and reinsert somewhere else. They'll take flanking information with them to the new location, and they might insert themselves inside genes, disrupting them. While this might seem a little bit strange, most of your genome is actually composed of dead transposons, remnants from previous viruses. Most of these are silenced by the genome through methylation. This can prevent them from jumping. This is also one of the things that causes something called hybrid dysgenesis. Have you ever wondered why a lot of hybrids are sterile or lethal? What triggers the jump, Sarah? Is that the question?
Yes.
So it depends on the type of transposon. So most transposons in your genome are silenced, but some of them can be activated when you, for example, are affected by a different virus. Some viruses contain transposase, which allows them to insert in your genome and then remove themselves. When they make transposase, this can activate other transposons in your genome. So even if the virus isn't causing any problems itself, it can move from one location to another, an older replicant. There are also different environmental effects that can cause transposons to jump, and hybrids have problems with this as well. So for example, all genomes contain large numbers of silenced transposons. These silenced transposons require special regulation. When you create a hybrid, some of that regulation is lost. Yes. So part of what your genome is doing is it's going to hold on to pieces of old viruses. It uses those pieces of old viruses to recognize new viruses. The parts of your genome that help fight other viruses also help silence transposons. So they work together, both to silence the pieces that are already present in your genome and also to fight new ones that look similar. Because different organisms are exposed to different viruses and have different transposons hidden in their genomes, they have different regulatory processes available. When you make a hybrid, some of those processes are lost, which causes transposons to now be unregulated and jump. This is one of the reasons that hybrids are frequently sterile or lethal. Too many jumps means too much damage. This is also another way that bacteria can become resistant to antibiotics. Because transposable elements can carry genes with them, a transposable element that contains an antibiotic resistance gene can easily jump from the genome to a plasmid and vice versa. This means that new plasmids can carry resistance genes even if they didn't previously. And those can be spread to other bacteria through conjugation and then subsequent movement of the transposon. I'm not going to ask you about the specific mechanisms that transposons and viruses use to integrate into the genome. But I want you to understand that these are naturally occurring things that can move genes from one organism to another and do so all the time. We can see evidence for the movement of these genes in many organisms, even when we don't expect to see them. The other thing that's interesting about transposons is they create unstable mutations. Because they move, some cells will have a transposable element in one location, and other cells in the same organism will not. If you've ever seen an example of a flower petal or a corn that has spots like this in its pigment, these spots are caused by reversion of a transposable element. Each one represents the transposable element jumping out of the gene is disrupted. Some of the cells will have this happen, and most won't. And that gives this partial mutation phenotype. So all those really pretty corns that you see that have all those lovely speckled kernels around the fall time, this is why. They contain transposable elements that are sometimes able to jump. And certain types of corn are worse at suppressing jumping than others. So some have more spots. It is an evolutionary tool. So being able to transfer genes between organisms is really helpful, even naturally. Let's say, for example, you're a herring and you live in an ocean and you need to have resistance to something, and another fish nearby has developed that resistance. It's a lot easier for you to get the resistance from the fish that lives with you than to develop it yourself. So if you could get that gene from them, it would really help you. Organisms want to be able to take advantage of the positive benefits of transposons and viruses, but they also want to prevent them from being able to jump and hurt them. So it's kind of an arms race of sorts. How do you use something and let it happen without letting it hurt you too much? All of this gets us to this idea of what is a genetically modified organism. The exact definition of a genetically modified organism and what constitutes genetic engineering varies, and there isn't a good consensus on what that actually means. Most of the time, this does not occur naturally by mating or natural recombination. But that means that horizontal gene transfer would count as a genetically modified organism, since it isn't through vertical inheritance. However, most of the time when laymen talk about GMOs, what they really mean is transgenic organisms made in a laboratory. So what's the difference between corn where the transgene jumps naturally or corn where we jump the transgene on purpose? I don't know. There are several steps to making these genetically modified organisms, and some of them rely specifically on these natural types of insertion, while others rely on more complicated tool process. Generally, creating a genetically modified organism is a multiple step process. The first step is that you need to isolate the gene you want to insert into the host organism. This means you have to find a copy of the gene somewhere else, or you have to make a version of the gene. Once you have that gene, you want to combine it with ways to be able to follow it. If you don't know where the gene is, where it's inserted, or where it's going, you can't check up on whether or not it's doing what it's supposed to be doing. So you need to mark it in some way. You also need to make sure that the gene is expressed. We've talked about how genes require different elements like promoters and enhancers to be able to be expressed. Different organisms have different requirements. So just taking a gene from one organism and putting it into another one won't work most of the time. You have to think about what needs to be present if you want it to be expressed. The most common genetic markers that we like to use are things that are really easy to see. So for example, this is a transgene that's marked with GFP, a green fluorescent protein. Any place that the new DNA is incorporated will now glow green. This allows us to tell that 2 of these mice have taken up the transgene and 1 hasn't, and also where that transgene is being expressed. We can use multiple transgenes at once by taking advantage of different types of markers. For example, we have other colors we can take advantage of. We can also use basic genetic markers like coat color. For example, this is a chimera. This chimera is a transgenic organism created through multiple cells. The cells that are expressing some of the traits are brown. This allows us to show which cells within the organism are still expressing the original wild type traits, and which ones are expressing the new traits in the white areas. The most difficult part of creating a genetically modified organism is the last part, inserting the isolated gene into the host genome. It's not that difficult through PCR to find a gene you want to do, make enough copies of it, and add whatever elements you might want. But it's really difficult to convince living host cells to take up that DNA and actually use it. So that's usually the limiting factor. The first step, creating that gene, is usually referred to as cloning. Yes, there are steps engineers can take to prevent silencing of inserted genes. One is to make sure that the gene looks like a gene from the original organism. The other is to make sure that it's expressed in a way that the cell won't find threatening. So what happens sometimes when you insert genes is that they end up overexpressing, or they end up disrupting something that's important. And so the animal will try and silence that in order to remove this kind of disruption. So you can prevent this kind of silencing by controlling where the DNA inserts, so it's not in something that the organism wants. You can also do it by making it look just like one of the organism's genes, making sure it's not overexpressed, making sure that it's not hurting anything. The biggest one is controlling where insertion happens. And that's actually not as simple as it sounds. All right, so we are going to talk a little bit about how to do that, but first we're going to talk about cloning. And that's because this first process, the process of isolating and making your gene, that's cloning. A lot of times when we talk about cloning, we're thinking about making identical versions of organisms, like an identical twin. But in molecular biology, that's not what cloning generally means. Cloning is amplifying specific pieces of DNA in bacterial cells. So basically what we mean is we need to make millions of copies of identical things because we need a ton of DNA if we're hoping some of it will happen to get into the cells. This usually involves something called a cloning vector. That's replicating a DNA molecule attached to a foreign DNA fragment that we're going to put in the cell. So something that helps the bacteria make a lot of copies really quickly. We already talked about this idea that bacteria have plasmids or extra chromosomes that aren't part of their genomes. So that's what we can use most of the time for cloning. Cloning vectors generally have an origin of replication, which allows them to be replicated inside the bacteria. No origin, no replication. They also tend to have restriction enzyme sites. And we've talked about what restriction enzyme sites are for before in fingerprinting. These are sites that cut specifically. So for example, if we were to expose this plasmid, the restriction enzyme EcoR1, it would cut right here only. This allows us to open the plasmid and add new pieces of DNA exactly where we want them to. It also needs to have some kind of selectable marker. We want to make sure that we can force the cells to have the plasmid. Otherwise, because plasmids are transient, the cell will get rid of them. Usually, the selectable markers are antibiotic resistance. But sometimes it's other things. This allows you to grow the bacteria specifically so that it always has the plasmid and the plasmid cannot be lost. Once you have a vector like this, of which there are several you can choose depending on what you want to do, you need to put your foreign DNA into the vector. You're going to make your foreign DNA using PCR. So you go to the organism that you want to get the gene from. You use PCR to amplify a piece of DNA from that organism that you want to insert. You also can use PCR to add restriction sites. These probably don't naturally occur wherever you want to get this gene from, but you can add them. Once you do that, that means that when you expose your foreign DNA and your plasmid to the same restriction enzyme, they will complement with each other. So the sticky ends of the cuts will stick together, and your foreign piece of DNA will be part of the plasmid. You can then use DNA ligase, like you would during normal replication, to force the pieces together. Now, your foreign DNA is part of a plasmid that can replicate inside of a bacteria. So there are a lot of processes we've already discussed that go into the basics of cloning. We can use PCR both to get the gene and to make changes to it. I'm not going to go into details about how you use PCR to make changes, but you can. You can force changes into DNA by using very special primers. We use restriction enzymes to create sticky ends because they cut at very specific places. So we'll always get complementary pieces. And then we use the basics of replication such as ligase, and then DNA polymerase itself, to connect things together and replicate them. Most of the DNA technology that's used is always a combination of things that we've already talked about, which is why those core things were so important in the first place. Once the gene is cloned into your vector, now the trick is getting it into the cell. And that's the hardest part. There are a lot of choices. Some of them are more similar to naturally occurring transfer than others. For example, If you just want to move it between bacteria, you can use bacterial transformation or conjugation. You can use viruses to insert your vector into other organisms. You can also use physical methods that just put a lot of DNA into the cell, hoping some of it will be incorporated. Things like gene guns or microinjection. And there are some chemicals that can help you do it as well. But all of these rely on the host cell taking up and incorporating the desired gene into the genome. The likelihood of that happening is different for all the different techniques. And it's also different based on the organism you're trying to put the gene into and its defenses, and the cell type you're trying to put it into. Now, I know that you're not going to remember or think about all the possible ways you could insert DNA, but I want you to understand that getting the DNA in is the hard part. There are lots of options, but that at the end of the day, if the cell doesn't incorporate DNA, then none of them mattered. So we might think that it's easy to create a transgenic organism, but it isn't. It can take years to get just the right insertion that does what you need it to do. What if it inserts so that it's not expressed? What if it inserts in the wrong cell? What if too many copies insert? There are so many things that can go wrong that the process is screened extremely well. And it's not something that you can do overnight. If it's so difficult to do, why bother? Well, this type of basic bacterial transformation is actually used a lot in creating drugs and other therapeutic systems that are used by humans. The classic example is insulin. Insulin is something that most humans need in order to be able to regulate their blood sugar. Original sources of insulin were donor blood, pig insulin, both of these have disease issues. For example, if you're trying to get insulin from donor blood, you have to be able to get the protein out without giving anything else from the donor to the patient. If you're trying to get enough insulin from a pig, you also have to kill the pig in order to get enough blood. So this can be very costly. However, if you can make a bacteria produce insulin, you could produce an infinite amount with very little work or space required. Most insulin is now made synthetically in bacteria. In this case, the human proinsulin gene is inserted into the plasmid, just like we talked about. That plasmid is then grown in a lot of bacteria. Each bacteria will produce a lot of insulin, and that insulin can then be purified from the serum the bacteria live in. You can grow an infinite supply. You don't have to worry about transmitting human diseases or other things between organisms. In fact, a lot of things that we use today for medicine are produced this way. Here are just a few examples. Some of them have replaced previous animal or harvested samples from humans, such as human growth hormone, which we used to get from harvested cadavers, human insulin, which we used to get from cows and pigs and blood transfusions, follicle stimulating hormone, which we used to get from postmenopausal women's pee, and factor 8, which we used to get from harvested factor 8 from donor blood. All of these are much easier to produce and harvest cleanly from bacteria. Other types of things that we use, such as the human recombinant proteins, are only produced in bacteria and not able to be, at least at this time, received from other sources. Without the use of these transgenic bacteria, these would not be available to treat the disorders that they are for. We also use this process to make enzymes and other large quantities of proteins for industrial use. For example, anytime you see a product that says that it comes with enzymes, those enzymes are probably produced in a bacteria. This includes many enzymes that are found in detergents, in the production of clothing, food, and animal feeds, and some biofuels. Most are produced either in transgenic bacteria or yeast. So while we might think about transgenics as only being something that helps us medically, we actually do use this to create large quantities of proteins very cheaply. In this case, the organism itself is not used for the treatment or the product, only the protein that it's making. This is one of the things that makes this particular type of transgenics a little less sticky when it comes to the question of should we do it and is it dangerous? Since it's only the protein, the source of the protein shouldn't matter. Examples of proteins that we actually do make in order to do things like laundry detergent, if you've ever wondered why that's gotten better, are amylases, lipases, proteases, and cellulases. When you look at detergents that say that they have enzymes to take care of these different stains, these are the enzymes that they mean. And these enzymes are coming from cells. We also use this process to make transgenic animals. Sometimes this is more effective because of the way the protein has to be folded or treated. And there are ways to make transgenic animals to treat diseases as well that don't have to be killed. An example of that is transgenic goats. Transgenic goats and other mammals can be made to produce drugs or proteins in their milk. In this example, we're adding an antithrombin coding sequence to a goat. This sequence is normally found in humans. We splice the sequences together, and then we transform the cells. Most of the time, this is through microinjection, injecting tons of embryos with a lot of DNA and hoping that they will incorporate the DNA in the right place. Those cells are then fused with an enucleated egg so that they'll grow into a full embryo, transferred to a surrogate female, and then we hope that the babies will express the gene we want. But getting them to do that can take a long time and many rounds. Once we have founders who express the gene, we mate them to find ones that are able to pass the gene on. Now it can be transferred through vertical inheritance and won't be lost. In this case, the goal is to have the goat produce transgenic milk. These goats can produce up to 3 kilograms of this product per year. And the reason that's important is because this is an anticoagulant. The main purpose of this antithrombin is to prevent blood clots. Individuals who have this genetic disorder tend to have blood clots that form randomly inside the body. One goat can produce as much of this material in 1 year as 90,000 blood donors. Because the protein is purified from the goat's milk, it also isn't directly ingested in any other way, making it less problematic than some other types of GMOs. Are there any questions about this process so far? What is a transgenic organism? How is that different or similar to a GMO? What you need to do to make something a transgenic organism, and why you might want to do that? Now, one of the things you've probably noticed is that none of this is in humans. Getting DNA into host cells is actually really hard, like I said. Cells don't generally like having foreign DNA shoved into them. And when you're working with people, it really matters to you whether or not that delivery is going to work the first time, and whether or not it's going to cause problems for that person. So if you're trying to use this type of transgenics to fix a human disease directly, there are a lot more safety concerns. We don't necessarily care what happens to our transgenic goat as long as it's reasonably healthy and it makes the protein we wanted. But we can't have the same opinion about how this influences a person. The different ways of delivering DNA into an organism all have problems, and these problems are bigger when the fate of the individual organism matters. For example, virovectors can be used in vitro, ex vivo, and in vivo, but they often cause insertional mutagenesis. We talked about how naturally viruses can insert inside genes, disrupting them and causing cancer. That can still happen when you use a virus as a method of getting DNA into a person. They also tend to have limited cloning capacity. Viruses are small, so the gene that you put inside has to be small too. And because they are viruses, some people are allergic to them, or they can elicit large immune responses. So there are safety concerns. Physical deliveries can be used in vitro and ex vivo. That means that you can't use them on a living person. This is because they tend to cause a lot of cellular damage. So if you use them, they can create too much damage that the individual might not recover from. When these are used for gene therapy, they tend to be used on tissue that is removed, then grown outside of the person's body and transplanted back in. This gets around the damage problem, but it means that you can't necessarily use it on someone. And then chemical delivery methods are just inefficient. They're unlikely to get the genes where you need them to go. There are lots of different gene therapy methods that have been tried. Your textbook has a list of a few of them, the most common ones, the advantage of those and their disadvantages. And while I don't expect you to remember specifically what their advantages and disadvantages are, I want you to understand what some of the problems with trying to get DNA in are. For example, viruses tend to be really efficient, but retroviruses tend to insert randomly. And that means that they have a risk of disrupting genes or creating new wild-type viruses. Adenoviruses are helpful for transferring to non-dividing cells, but they tend to be bad for the immune system. Some work really well, like the adeno-associated viruses, but you can't put big things in them. Some of them can insert into the nervous system and don't cause immune reactions, but you can't put big things in those either. Some are awesome, but super dangerous. Good at targeting, good at putting things in, put anything you want, but also it's the same type of virus that causes HIV. So, there's a lot of issues with all of these. And that brings us to CRISPR. CRISPR is a new process for trying to edit DNA. The CRISPR-Cas9 genome editing system allows you to insert specific changes at specific locations. This helps get rid of things like off-target effects, where you have an insertion where you don't want it to be, which is one of the main problems with viruses. So what are the parts of the Cas9 system? There are three major things that you need in order for Cas9 to be effective. You need a single guide RNA, the Cas9 protein itself, a targeted piece of DNA that has a PAM sequence nearby, and you have to be able to get all three of these things in the same place. When the three of them are together, the Cas9 protein opens the DNA. It does this at PAM recognition sites and places where the guide sequence matches and is complementary to the DNA. This allows you to create specificity. The Cas9 system will only open the DNA where both the guide sequence fits and a PAM sequence is located. When this happens, the Cas9 system creates a cut in the DNA at this specific location. DNA repair mechanisms want to close cuts like this. You can take this to your advantage by supplying donor DNA. Donor DNA is similar to the DNA at the location of the cut site. When the DNA is repaired, the donor DNA can be incorporated into the cut location. This allows you to replace a gene with a previous gene or a change to a gene. It also allows you to remove a gene if you do not provide donor DNA through end joining. So CRISPR can be used both to make edits to genes, replace genes, remove genes, and add genes in a very specific way. Because of the high specificity and the fact that it does not rely on the use of viral vectors, although they are still sometimes used, CRISPR has made it farther in a lot of gene editing processes. Some examples, mostly recently, are as follows. CRISPR-Cas9 has been used to treat sickle cell disease, replacing mutations in the gene responsible for this disease with wild-type versions of the gene. The first trials for this were started in 2019, and successful patients have been developed since then. It's also been used to treat cancer. Because cancer is frequently caused by mutations, you can edit cancer mutations to remove the growth factors. We've used it to do things like treat type 1 diabetes. There's currently a clinical trial for that as well, where it seems to be promising from this year. And we can also use it to treat other disorders. For example, just this month in November, there's been some success treating angioedema, which is a type of inflammation and swelling, using CRISPR. That doesn't mean that everything that CRISPR is used for has been easy or necessarily a good idea. You may remember that in 2018, a Chinese scientist used CRISPR to modify infants. These individuals needed a resistance gene to protect them from HIV. And while there are non-CRISPR methods in order to prevent HIV transmission from mother to child, he decided to use CRISPR to do it instead. We also know that there are individuals who have died during CRISPR trials. One recently, just from this month in upstate New York. Most of the time so far, when individuals die, it's because of the method used to get CRISPR into the patient. For example, in a previous trial, the patients who died all experienced cytokine release syndrome, which is a huge immune response. Some of the patients were not able to be stabilized afterwards. So there are some downsides to CRISPR too, but it might have a lot of promise. So what is good about it? The change in CRISPR is mostly not random. It should only occur where the guide RNA says it should, and where there's a PAM sequence. It's efficient at inserting the donor DNA. As long as you can get the CRISPR there, it is going to make the change. You don't have to hope that the DNA will be incorporated. It's cheap, and it's really easy to design. Guide RNAs are easy to make with PCR. There's no size or design limit. You can make something as large or as small as you want, and you can use it in living tissue, whole people, and any organism. And if you want to edit RNAs, you can do that too. It's not only for DNA. So it has a lot of benefits over other methods. But there are downsides too. It still needs improved delivery systems for in vivo editing. If you want to make changes, you have to be able to get the system there. Different treatments in different cell types need different deliveries. And sometimes viral vectors are still used, and these can cause immune problems for the patients. You're limited in where you can make changes. There has to be a PAM sequence nearby. If the PAM sequence is far away, you need a huge guide RNA to get to where you need to go. It has limited cutting power in certain locations, although there are some improvements to help with this. And even though it's much better at being specific, there are still risks for off-target mutations. There are lots of places in the genome with the same sequence. So this gets us to a bunch of questions about, should we do it? There are potential problems. In general, most people are consistent with their feelings about somatic cell therapy. Somatic cell therapy is when you change the expression of genes in the body of an existing person. These are most likely to be used for clinical development, things like treating actual disorders that the person in front of you have. And most people don't have large ethical concerns about this because the person can easily consent. And somatic cell therapy is not transmissible from one person to the next. But when you start getting into germline therapies, that's where there are more ethical concerns. For example, if you make changes to the germline, these changes would be passed from one individual to the next. There are very inconsistent guidelines about what is and is not allowed, and there's very limited research on how this is going to work out. One of the reasons that this is a little more controversial is like this. Let's say that your intended consequences for CRISPR are to make changes to a gene that is expressed during early development, so that this individual won't have this disorder. Your goal is a live birth that doesn't have this disorder and elimination of the disorder from the gene pool. Or it might be research on how this disorder develops. If those were the only things that happened, this might not be so bad. But if there is an off-target effect, this could lead to developmental defects and cancer risk in the child, even if the repair occurs. And that mutation may enter the gene pool too. So while you may remove one, you may introduce another. You also might create cytotoxicity. This is an issue where you create changes in the embryo that cause the embryo to be unable to develop, even though it should be able to develop normally. Or you could have a situation where the insertion is partial. So the person still has the disorder, but not everywhere. Is that sufficient? So while in a perfect world, this might seem like it's pretty straightforward, in reality, there are a lot of possible pitfalls. When you talk to people about their feelings about these types of changes too, you can see that there is a big divide between somatics and germline changes, and also what those changes are used for. For example, in the US, most people are comfortable with using these types of technologies to fix diseases or conditions that babies would be born with, or that they might develop over time. But most people are not comfortable using them to change things like intelligence or appearance. This raises questions of things like, is it ever okay to design a baby? Is it ever okay to select a child based on their genes? Is it okay to avoid having a child with a disease? Then is it okay to edit a child's genes? These are not questions for which there is a clear and perfect answer. All of them are going to be subjective and based on where you're from, your own personal beliefs, what it is that they're trying to change, and why. These also connect directly to ideas about genetic testing. For example, in the question of, is it ever okay to avoid having a child with a disease? Genetic testing allows us to do just this. It also allows us to determine which child we might want based on genes. If we can do these things through detection, then we could make changes to prevent the disease or to get the trait we wanted. So while we've been talking so far about making transgenics, how to make the change, we also need to think about, should we be able to find the change in the first place? Genetic testing also has multiple different facets. We use it to test for fetuses or early embryo development. We use it on newborns. We use it as a predictive measure to look for pre-symptomatic individuals. We look at it for carrier testing and also in direct-to-consumer genetic tests and products. In prenatal genetics, the goal is to detect the risk of a fetus having a disorder, generally a single gene disorder. Why might we want to do that? Well, we might want to avoid passing that disorder on to our offspring, or we might want to avoid bringing a child into the world who might die shortly after birth. To do this, we can take some of the fluid from the some of the amniotic fluid, to look at whether or not those specific mutations are present using PCR and DNA analysis. We can also get some of this information now from maternal blood without having to interfere with the fetus at all. However, testing comes with its own issues. What if the outcome indicates the fetus has a disorder? Your options are then to prepare for the healthcare needs of that individual, or to think about whether or not you want to have an elective abortion, or you may need prenatal or newborn surgery to correct the problem. If you can't have an abortion, or can't have the surgery, or can't afford the needs of the individual, does knowing about it help you? Hard to say. The same thing is true when thinking about in vitro fertilization. In vitro fertilization, genetics are done on embryos prior to implantation. This can be used to detect disorders so that embryos containing those disorders are never implanted. But that means that individuals can choose whether or not they want to have children with various different traits. It also allows individuals to choose whether or not they want to have a boy or a girl. Is that something we should allow? Some countries have banned the ability for parents to choose the sex of their embryos through in vitro fertilization, but the US is not one of them. So if you were to go through this process, you can choose which type of embryo you'd like. You can also select against other traits if you know you happen to carry them, but different states have different rules about what's allowed. If you want to do something that your state doesn't allow, you could simply go somewhere else for your IVF. So is there something we should do about that? Hard to say. Newborn screening is another thing that we tend to do. This is a lot less controversial, although there are still people who are concerned about it. That's because, in general, newborn screening is about preventing problems. The effect of not screening in this case is absolutely worse. And let me give you some examples. One is PKU. PKU is a disorder that affects 1 in 10,000 individuals, and it's controllable by changing diet. When PKU is not controlled, it results in severe mental retardation and other birth defects. But if the diet of the infant is controlled from birth, they will be normal. In fact, that's true for most of the things that newborn screening checks for, like congenital hyperthyroidism, adrenal hyperplasia, galactosemia, PKU, and G6PD. All of these can be treated before birth or shortly after to prevent any of the negative consequences. In this case, not testing seems to be less ethical. What about if you're a carrier for something? In that case, maybe you want to know, someone in your family has this disease. Do I carry it too? Classic examples are things like cystic fibrosis, Tay-Sachs, sickle cell anemia, and Huntington's. But this brings up the question of, what if I am a carrier? Who do I have to tell? Do I need to tell my spouse? Should I tell my brothers and sisters? Who needs to know? This type of testing is generally recommended and is offered by insurance to most individuals who are interested in having children. But you should consider what it means for you before you have it done. The other concern, of course, is that testing or screening for genetic disorders may reduce the likelihood that those people will be born. And different groups of people do not want that to happen. For example, if you're selecting for whether or not your child will be blind or deaf or have Down syndrome, people who are blind, deaf, and have Down syndrome can live very fulfilling lives. Is it okay to decide that that child isn't the one you want to carry? Sometimes it makes sense, like for Tay-Sachs, which is lethal after 2 years. In this case, we can show that doing this type of screening does allow us to reduce the incidence of these deaths. It's 90% since 2000. But we also can use this to reduce other people. Should we? There are a lot of other complications for DNA testing as well. Things like you may discover that parents are not who you think they are. Or this could be used to prevent having a child with an undesirable trait, whatever that person thinks that is. Law enforcement can use testing done by someone else to determine whether or not you might be a criminal. Indirectly, without a warrant or consent through search and seizure. The information that you learn could become public. It could disclose your disease risk to your insurance provider or employer, or your disease status or risk to your sexual partners or spouses, or could subject you to individual bias, exclusion, or discrimination in various places like education and the workforce. So even if you do DNA testing, who are you obligated to tell? Who has a right to know? What can that information be used for? As a doctor and someone thinking about potentially being a PA or working in medical fields, you also need to think about, what does the patient want? Is there any condition that they would not want to know about? Conditions that occur later in life tend to be scary. Patients might not want to know whether or not they're going to develop Huntington's disease because they might not want to be afraid. Or they might want to know so that they can plan their time. And every individual is different. So even if you think someone should be tested for something, it's important to keep in mind their own feelings about that information and how difficult that information might be for them to deal with. For example, fatal versus chronic versus progressive treatments. If you know that it's fatal and untreatable, is it worth knowing? Hard to say. If it is treatable, it's probably more worth knowing. If you can diagnose early, is it easier to treat? When is it going to happen? So when you're thinking about all of the different parts of using this type of technology, making different organisms that can behave in specific ways, or trying to determine whether or not an individual has a trait or what you might do about it, it's important to keep in mind, should you? What are the limitations? How could this be misused? What could go wrong? And what does the patient want? How do we help the patient? It isn't always by knowing more. Are there any questions about using testing, about CRISPR, about things to think about when it comes to the ethics of these processes? Or any comments you might want to make, or questions you might want to ask your fellow students?
Can you reiterate the difference between like the GMO kind of gene editing and CRISPR again?
Sure. CRISPR is a type of GMO.
Okay.
Okay, so essentially a GMO, the term GMO means almost nothing. So GMO just means that the organism has some genetic change that was not inherited. Whether or not that refers to specifically lab-made changes or any non-vertical change depends on who is writing the paper. So when people talk about GMOs, you want to look really closely at what their definition is, because this is somewhat subjective. When we're making lists of what constitutes a GMO, this is things like a crop that has a gene inserted, or an animal or bacteria that makes a drug. So they might have a human gene. This would also include that lady who was cured of her sickle cell. She's a GMO now. Right, so CRISPR is a technique that is used to produce a genetically modified organism. So we talked about there are the three major types, the types where we use DNA that's in the environment, the type where we use viruses to insert DNA, and the type where cells just trade DNA. CRISPR falls into this first category. Because we're providing a kind of special donor DNA, and we want that special donor DNA to be inserted. And so this is kind of a delivery method. It's a way to get that DNA to a specific location and try to get that DNA to insert in that location only. So while GMOs refer to all organisms that have these types of changes, CRISPR is just one technique for creating a modified organism.
And so do a lot of the benefits of CRISPR come from the use of RNA targets? Or is that kind of just like an aspect of the method?
So the guide RNA is the way that you direct where the cut will happen. So if you want to put the DNA in, you have to make a cut around the gene you want to change so that that can go in there. There is no difference between a transgenic organism and a GMO. That's semantic. All GMOs are transgenic organisms. The only difference depends on your definition of GMO. So if your definition requires lab made, then natural transgenics doesn't count. If it's any non-vertical transmission of genes between unrelated organisms, then natural transgenics does count.
Mm-hmm.
Does that help me? Yes. So this is one of the things where, like, so people will talk about, oh, GMOs are scary, oh, GMOs do this, oh, GMOs do that, but the term GMO doesn't mean a lot. You know, it could refer to an organism where you jumped a transposon and now the organism has this trait. You didn't add anything, you just moved the transposon. So maybe you exposed the plant to a specific drug that activates this one transposon set, and then it happens to jump in a good location. Well, that's a genetically modified organism. You didn't get that change through breeding, so now you have to label it as a GMO. It's super general, doesn't mean a lot. So it's okay to say things like GMOs are dangerous because some of them are. But you have to be more specific about what it's doing. So like the ones that tend to be dangerous are ones that add allergens to plants by accident or create byproducts that are toxic. So one of the classic examples is a corn GMO where the pollen is toxic to other plants because of the resistance gene that's present. So it's not toxic for you to eat the corn. You're not eating the pollen, it doesn't do anything. It's easier for the farmer to grow the corn because it's not affected by this disease anymore. But other crops that live around the corn can be influenced, and other plants that are not part of the crop system can be influenced. So that has a bad environmental effect, right? So when we talk about GMOs being good or bad, it's more about not always understanding the consequences of what you've changed. Sometimes the consequences are that it makes more vitamin K now. Awesome, cool. There's not really a big problem with that. But sometimes you get off targets that can be dangerous, or you get environmental effects that can be dangerous. So it's a lot more complicated than that. So CRISPR does not use restriction enzyme sites. It uses the guide RNA and the PAM to determine where the cut happens. So CRISPR itself is going to make that cut. Restriction enzymes, they look at sequences of DNA and they cut where the sequence is the same. So it's related. They're both using a consensus sequence, but CRISPR uses the guide RNA to determine that. The protein doesn't do it itself. It just goes to the RNA and the RNA provides the location. If it detects both, that match in the RNA and the PAM sequence, then it will cut. Let's see. CRISPR can be used to fix inheritance. So this goes back to this idea of where are we making the edit? So the biggest thing here, let's get, need a better place to write. All right, let's get this. All right, so when we're thinking about inheritance, that's the difference between somatic versus germline transgenics. All right, so when we're treating a disorder, we want to think about a few things, and this goes for the topics that you guys are working on as well. Where is the problem? So for example, let's say that the problem is blindness. And there is a CRISPR clinical trial that is effective. Yes, whether or not the gametes will be modified. In this case, there are two ways to accomplish that, Sarah. So you can either make changes to the stem cells that make the gametes, or you can make changes to an embryo and hope that the cells that will become the cells that make the gametes are changed, or you can make changes to the gametes directly. Right? There are lots of things that you can do with that. But it's about where. Like if we want to cure blindness, we really only need the CRISPR to get into the eye. So they actually have a CRISPR delivery system that's eye drops that will deliver the CRISPR system only to the eye. In that case, the likelihood of off targets in other parts of the body is basically zero. You could get off target insertion in the eye somewhere, but you're never going to pass that trait on. It's localized to where you added the CRISPR, just to the eye. So when you use things like CRISPR in adults, this is not likely to be passed on. You would have to deliver it directly to the cells making the gametes. And in women, that's going to be impossible because the eggs already exist. And in men, that would have to be specific cell subtypes in the testes. So it's very unlikely that any CRISPR treatments in adults, existing individuals, would ever be passed on to their offspring. It runs into a problem when you start making edits to embryos. So when you have a developing blastocyst, for example, and you're testing this blastocyst because you're going to do IVF and you fertilize this embryo and you want to see whether or not it has this